Hey guys, today I'm going to show you how to draw an animal cell. As you may know, animal cells are eukaryotic cells and unlike plant cells do not have cell walls and chloroplast. So let's get started. First, let's start with a simple circle or oval for the cell membrane. The cell membrane of an animal cell is not a perfect circle, so don't worry too much about making one. Next, erase a small area from the cell membrane and push a small circle inside the cell. This is the pinocytic vesicle, which allows extracellular fluid inside the cell for digestion. Remember, the animal cell membrane is semi-permeable. Draw a circle inside the cell, which is the nucleus. Add a darker, smaller circle inside the nucleus, which is the nucleolus. Add a thin layer around the nucleus, which is the nuclear membrane. The nucleus, as it controls the cell and its functions, contain chromosomes and DNA. Draw a squiggle inside the nucleus to show the chromatin material such as DNA and protein. Animal cells also have nuclear pores which allow materials to go in and out. So add little dots around the nucleus as well. Next, we will draw a ribbon-shaped endoplasmic reticulum. Animal cells have two types of ER, smooth ER and rough ER. To make the rough ER, place dots on the outside of the ribbon shapes on one side of the ER. These dots represent ribosomes. Let's not forget to label our organelles as we keep drawing. Next organelle are the lysosomes. These are the garbage bins of the cell who eat up and digest all the unneeded material in the cell. They are oval in shape and look like capsules, and you can place them in the cell among the organelles. Next is the powerhouse of the cell, the mitochondria. Draw them by making 2 or 3 large rod-shaped ovals in the cell but outside the nucleus. Each mitochondrion should contain some squiggle and ridges inside. This shape represents the mitochondrial cristae helping mitochondria with its function. We now draw another organelle, which is the Golgi body or the apparatus or complex. It usually looks like a set of 3 curved dumbbell-type shapes that are cylindrical at the center and bulbous at the end. Do not forget to capitalize Golgi since it is the name of the discovering biologist. Now, add 2 to 3 vacuoles, which are these empty compartments which is to transport materials outside the cell. Add the centrioles, which are basically a bunch of tubes lying together. Draw thick cylindrical shapes for the microtubules that are thin filaments. Add dots inside the cell but outside the other organelles to show ribosomes. Finally, all these organelles are suspended in a fluid called the cytoplasm of the cell. Lightly shade and label the cytoplasm. But remember to leave the vacuole empty. And voila, you now have a complete animal cell diagram.